and welcome back to our third and final day of Dateline Live. We're so happy that you've stuck with us this week and have decided to join us again today on Friday afternoon. We have a great lineup again today, a little bit shorter day today so that you can get your weekend started. But we again are just so grateful for everyone for being here. My name is Sonji Wilkes. I'm the Vice President of Policy and Advocacy at the Hemophilia Federation of America. And again, I just want to say welcome. We're so grateful that folks are willing to still do in virtual events, and we can't wait to see you in person in the next few months, we're hoping. So let's get today started with a couple of reminders and housekeeping type things, and then we'll jump into our panel. So as always, we like to thank our sponsors, and we really couldn't have done this event, our inaugural Dateline Live event, without them. So thank you to CSL Behring, Hemobiologics, Genentech, Sanofi Genzyme, Takeda, and Spark Therapeutics. We really appreciate their support. Also, again, it kind of sounded like a broken record, but those Zoom helpful hints. This session is being recorded, so in case you missed something earlier in the week or have to step away for a few minutes today, just know that those sessions will all be available on the HFA website within the next week or so. If you have any questions, need some technical help during the session, use your chat box. HFA staff is kind of on the back end of things here, helping answer and direct traffic as need be. So we're here for you. And then if you have questions of the panel or any of our speakers, please use the Q&A feature, because that's where they'll be looking for any kind of questions from the audience. And again, just know that it's being recorded so that so we can watch this at a later date. So with that, just a quick review of today's agenda. We're going to do some information about registries. We've got a great panel coming up that'll tell you about different registries, not only here in the United States, but across the globe. And then after that, Dr. Dave Clark, who has been just a phenomenal resource for HFA for many years and for the Coalition for Hemophilia B, will talk to us about emerging therapies and products that are in clinical trial right now. So stay tuned for that. And then I'll just come back and offer a few final words. I bet I get you out of here before 6:45. So we'll aim for that so you can get your weekend started. So I would like to welcome back Eric Iglooski from the Mary Gooley Treatment Center. Eric is a social worker and has been a moderator extraordinaire for us this week. So Eric is here again to moderate our panel on registries. And Eric, I turn the floor over to you. Just really appreciate your support, and I'm glad to have you.
Thanks, Sonji. Welcome, everyone. Appreciate you taking a little time out of your Friday to join us. This is a really important session, and I want to give you the lay of the land. As Sonji said, the title is "Get Counted: The Importance of Registries," and if our panel does their job right over the next hour or so, you're going to learn a little bit, or hopefully a lot, about how critical research and data is to our community, how it can lead to better treatments and health outcomes, monitor for safety, and help foundations like HFA and NHF learn and just do better at serving their communities and members. I can speak for myself, maybe a little bit for you. Again, as you heard, I'm a social worker. I'm not a doctor, nurse, or scientist. Research and data is overwhelming for me. It might be for you. I get confused about all the different acronyms. Thank goodness for the HFA session yesterday on acronym bingo. It helped me a lot. Our experts will be able to shed some light on things and help you come away with a better understanding. So as Sonji said, this is going to be a webinar format. You're all going to be muted, but you will have a voice, and I want you to use it. Please chime into the chat, connect with each other, and please do use that Q&A button, and keep my speakers on their toes. See if you can stump them, throw some good questions out there. And if you don't have some, I got a few up my sleeve as well. So, and just a fair warning to my speakers, I may interrupt from time to time if a good question comes our way. So I'll try to merge safely without bumping into anyone. And I am the timekeeper. So if we're on a roll, I do want to be respectful to the 5 o'clock sessions. So I may have to pump the brakes when we get close to 5, so just be ready for that. So without further ado, we're just going to go down the line. I'd like to introduce our first speaker, an old friend of mine, someone from NHF, Michelle Whitcomb. Michelle, can you introduce yourself a little bit? And if you could, can you tell us a little bit about what registries are and why they're important to our community?
I am more than happy to do that. And I'll bring up my screen. Thank you, Eric. Let me just do this real quickly. Doesn't look like it's showing anything. Let me try again. I'm sorry. There we go. Can you see it all right, Eric?
You're doing great, Michelle.
Perfect. All right.
We're there. We're there.
All right. I'm Michelle Whitcomb. I'm the head of research at the National Hemophilia Foundation, and I really appreciate that HFA invited me here today, as well as had this really important talk for Dateline Live on registries. First off, the question that I was asked to talk about is, what are registries and how are they important to the community? And we have several registries that we are going to all speak to individually. But as a general overview, registries are really important for the rare disease community. And this is an opportunity for us to gather standardized information about our population. When we say a group of individuals, it could be hemophilia, it could be von Willebrand's, other ultra rare diseases, and to really understand the journey that you have as a person who's affected with one of those disorders, we really need to gather information about it. You know, to know somebody is to know that one person, but when you have information that you're gathering from a multitude of people in a standardized manner, that really allows us to look for patterns, trends, and changes over time. And that's what registries do. It's a very powerful tool. And it allows us to really understand a disorder and understand the journey that you are experiencing as a person who is affected with a disorder. So the more people that are involved in a registry, the more information that we have and the more powerful it becomes, the more powerful the results are. As it says at the bottom of the screen, every person matters in a registry. So why are they important? Again, it's understanding the needs of the community and the needs of the people who that registry serves. In order to make that registry appealable to that community, you have to really get to the questions that are important. And we're going to talk about that as we go through this hour. Understand that not one registry can meet everyone's needs. And that's often a question that we hear. Why can't they just have one that does it all? And that really is difficult to do. And what I hear over and over again from all types of disorders, all types of registries, is that there is not one magic bullet that we can have as a registry that is going to answer all of the questions. So that's why we have different registries from different perspectives. And when you create a registry, you're really bringing together that entire community, looking at the patients, the caregivers, the clinicians, researchers in industry. And the purpose is so that we can understand again the people that we are serving. When you have a specific condition and when you ask the right questions, you are more likely to serve that population, and you're more likely to draw in people who are interested, as well as you're more likely to draw in researchers who want to design better studies based upon the information that they can draw from the registry. And so that's an important thing to remember also. The information that they see in a registry will help them as they start designing clinical trials and do further research that can help the people in the registry to live a better life and to have better treatments of the disorder that they are living with. So registries really connect that patient community with the researchers. It really allows the researchers that glimpse into what it is like, again, to live with a disorder. And while we're talking about registries today for inherited bleeding disorders, there's registries for everything out there. Muscular dystrophy, there's a registry on pregnancy. Just to understand what it is like to be pregnant and be able to answer some of those questions. So there's all kinds of registries out there. And what it does is it connects that population to researchers so that they can understand that population better and they can move forward with research that's going to improve the quality of life and the treatment for the population of the registry. So today, I'm going to start off with several registries: the Community Voices in Research from the National Hemophilia Foundation, which is a patient-focused registry; ATHEN, the American Thrombosis and Hemostasis Network; the ATHEN Dataset, which is another registry that comes from our hemophilia treatment centers and are provider-focused; and then the World Bleeding Disorder Registry. We're going to start right now with CVR, or Community Voices in Research. And Community Voices in Research is a community-powered registry. And we chose that because it is powered by the people who are affected with an inherited bleeding disorder. Specifically, and it's general to all inherited bleeding disorders. It's not just for hemophilia. It is for all inherited bleeding disorders, as well as their unaffected family members, because we know that it takes a village. Those people who are not affected but live with you and care for you walk that journey with you, and we want to understand what that journey is like also. So what this does is it allows us, again, that peek in the window to get that 360 view of what it is actually like to live with a bleeding disorder. And when I say we, it's not just NHF. This is open to researchers. And the researchers that have used CVR to date are Hemophilia Treatment Center physicians and researchers. We've partnered with ATHEN. We have had students who have used the data, as well as industry partners. So again, it's a community-powered registry. It's available to the community, and the information is available to the community. The information in it is self-reported information because it's direct from the patient or from the participant's voice. It's direct from them. It is all through surveys at this point. It starts out with an enrollment survey, and then it is followed by a baseline. And I'm going to show you the depth of the baseline information that we gather. And this is both for affected as well as unaffected persons. And then we have hot topics. So if there is something that's going around and we really want to get a real quick glimpse as to what is going on with that, it'll be just a 5-question or so survey. We'll send that out. And we did our first one when vaping was a big issue, and we wondered how that was affecting our community. When COVID first came out, we did a COVID survey with the community. And then we recently have done a COVID survey in collaboration with Germany, the UK, and with HFA. We did a pain survey and a logging surveys as hot topics. And pretty soon we're going to be having a core survey come out, which will be repeated year after year after year, because CVR is what we call longitudinal, meaning that it follows people through the years, again, looking for patterns, trends, and changes. And then we also have research surveys. So if researchers have a specific survey that they would like to send out, then we do that also. What the participant gets is in return is that they get that information back immediately in the form of a personalized dashboard. So they can see everything in a de-identified and aggregate manner, meaning grouped together. And they can see how they responded and then how other people responded to the same questions. We have educational resources available so that they can push out information about what it means to live with hemophilia. What does Tylenol or acetaminophen do to your body, and how do you need to watch for it? All different types of educational information is available. When it was Mental Health Month, we had a bunch of stuff on mental health. Joint Month in June, there's a bunch of stuff on joint. So that type of stuff. We also offer virtual advisory panels because we often get requests from people, from sponsors, that they would like to be able to speak directly to people who are affected. And so we have like this, in a virtual setting, we invite people who meet certain criteria. It's all anonymous. They are compensated for their time, and then we set up discussions so that certain answers or questions can be answered. And as sponsors are starting to develop their clinical trials, the FDA now really wants patient input into that design of clinical trials very early. And this is a way for us to get that patient engagement in clinical trials at an early stage. In the future, we're looking, after we have our State of the Science Summit in September, we're looking at developing a clinical trial finder, because we know how difficult it is for people to go through clinicaltrials.gov. And so if we can pull all of that information on clinical trials that are specific to inherited bleeding disorders into one format and offer it in an easy to understand way, that's something that we would like to do. As I mentioned, that we were going to show you just some of the information that we gather in our baseline, and you can see it here. And much of it is stuff that has never been gathered in other places. And remember, this is direct from your point of view. There's no filters. Nobody is saying, that's not exactly what I think that person meant, and so changing it a little bit. This is your voice. There's a lot of stuff in here, and those are reflected back into those health reports on your personalized dashboard. And this is shows again what, as a participant, you get. By participating, you get your personal dashboard, opportunity to participate in virtual advisory panels. A personal CVR record is where you have the opportunity to input your information that you would like. You can print it out and then take it to a healthcare provider. So if you're with a primary care or a dentist, that gives your personal record that you want to share, and it's in a one easy format. And there's a QR code right there if this is something of interest to you. And you can always go to www.hemophilia.org/CVR for more information or to enroll. And with that, I want to talk about ATHN, and I apologize for the blurriness of this because I did a screenshot on it. But ATHN is the American Thrombosis and Hemostasis Network, and we're going to talk about the ATHN data set. And ATHN, unlike CVR, ATHN is through the hemophilia treatment centers. And this is a data set that's available through the providers. So when you go in for your comprehensive clinic visit and they gather that information at your comp visit, they are putting, with your permission, that information into the ATHN data set. And that's where that information comes from. So this is a provider available data set. And the reason the researchers are gathering that, this is to improve improve our understanding of both the clinical, social, as well as economic issues that are affecting our families. Excuse me. where before we were talking about CVR, that was patient self-reported, this is clinical. So this is all of that clinical information that your lab reports and things like that might be of value to understanding treatments. They can understand the development of inhibitors and monitor trends. They can look at genetics. They can look at the effectiveness of treatments and also help in developing standards of care. And then when they're monitoring your therapies, they're being able to report back to the FDA as to whether there's any potential side effects that were not previously reported, or if this is an approved medication that is following the course that's expected. So that's the type of things that they use with the Athens data set. And this is the type of information that is gathered in the Athens data set. And you'll see sometimes there are similarities with what we talked about in CVR, but then again, there are different things too. And that's what's important. Again, we said you can't get everything in one place. So that's the benefit of joining several of these. Now, WBR is the World Federation of Hemophilias. It's their World Bleeding Disorder Registry. And when we look at WBDR, this is that global perspective of Athens. Athens is a participant in WBDR, and WBDR takes it to that global perspective. And again, with informed consent from the patient, the hemophilia treatment centers across the world gather that information. And again, I didn't mention this with Athens, but it is anonymous. It is reported out in a de-identified and aggregate manner. So it gives information about the type of bleeding disorder you have, the symptoms, the treatment, and it allows the countries as well as the WFH to be able to look for patterns, trends, and changes at a much higher level. So here's the information that is, well, I should say that this is not the information that is gathered from WBDR. It's similar to what you saw with Athens. But their goal is to be in 50 countries at least and have 10,000, if not more, people enrolled in this through their HTCs. And Athens participates in this. They send about once a year their information to WBDR to be downloaded. WBDR uses this information to help us get that global perspective of what it's like to live with a bleeding disorder, as well as to help promote information at a country level, to help promote registries at a country level, to be able to get access to care at an individual country level, all of those types of things. So this information is vital for us to have. And with that, I am done with my section and I'm ready to turn it over to, I believe, Mabel.
That's correct. Michelle, that was fantastic. Thanks for taking the time to cover all that. Mabel, you've got the mic now. Can you unmute yourself and open up that video?
I am.
There you are. So yeah, please tell us about you and your role with HFA. You're part of the home team here. tell us your role.
Home field advantage.
Exactly. And tell us about the registries that you're going to present about.
Sure. So I lead the research team at HFA, and I'm going to talk to you guys today about two different registries, the community counts and also the HFA research portal. And so I'm going to share my screen here. And you should be able to see. Okay, so you should be able to see my slides at this point. And excellent. Thank you for letting me know, Eric, that I managed that. So like I said, I'm going to start talking to you a little bit about community counts. And, you know, this is a partnership between Athens, which Michelle already introduced that organization to you, and the Hemophilia Treatment Center Network and the CDC. And these, I'm trying to figure out how to advance. Okay, here we go. So the Community Counts project is really focused on gathering data that they can use to learn more about patients with bleeding disorders and clotting disorders at HTCs. And so this is really focused on what happens at the HTC level. And their objective is really to learn more about inhibitors and other complications and how to reduce and prevent them, and really how do they improve the quality of life of people with bleeding disorders. So they collect different types of data, and one of them is the Htc Population Profile. And so this is really focused on taking sort of a head count of people with bleeding disorders and clotting disorders. And so being able to understand how many patients receive care within the HTC network and what does their demographics look like. And then they also have the registry for bleeding disorder surveillance, which is collecting much more detailed clinical information that helps them monitor trends, identify risk factors, and also rates of complications and high-risk populations that they can target for prevention programs, education, and other those types of activities. And finally, they also collect mortality reporting, which is really gathering information about causes of death of patients who have received care at HTCs. And this data has been collected since September of 2011. So we don't necessarily have data before that. So who can participate in this particular registry? This is really open to all patients who have hemophilia A or B, von Willebrand disease, any other rare bleeding disorders or platelet disorders, and some clotting disorders, so long as they're receiving care at a federally funded HTC, they're eligible to participate in this registry. So in terms of privacy, they talk about how the data is secure and confidential, that's covered under the HIPAA privacy rule. And patients, when they go to an HTC, they may be asked to sign a consent form. And so you can actually sign up for community counts at your HTC and consent that your data be shared in community counts. Some of the benefits to participation, really here community counts is focused on improving medical care and health outcomes. And so that's really how you benefit from participating in this, that the information that is collected is really used to inform and improve and guide how clinicians treat patients with bleeding disorders. And so participating in this, it may not be an immediate return, but it's really about helping to shape the future of medical care for people with bleeding disorders. And that's a pretty powerful contribution. And also, you help CDC sort of monitor trends in bleeding disorders over time. And, the CDC works in gathering a lot of this information about people with bleeding disorders, and they look at the size of the population, they look at medical complications, at, medical issues throughout the lifespan. And they also use that data to develop educational programming and educational tools. And I can tell you, having worked right now on a CDC cooperative agreement, that a lot of the data that is collected through the community counts, we use it to make decisions about what programming we should engage in and how to allocate the resources that we have. So now I'm going to switch gears a little bit and talk about HFA and what we're doing at HFA. And so we have a community research portal, and our research portal is really a platform that we use to collect data on the bleeding disorders community. And our objective is really to figure out how we can better serve the needs of our patients and their families. And so we really have whereas sort of what Michelle talked about earlier, they're focused on how do they support researchers in clinical trials. We are really using our data to understand what our community is going through, what our patients' experience are, and what their families go through, and then how can we use that information to then turn it around and create tools, education, or other types of interventions that can support our families and help them navigate this process. So why do we have a portal to collect data? So we actually set up this portal a few years ago. We have support from Genetic Alliance. Genetic Alliance received a grant through Robert Wood Johnson Foundation, and we partner with Genetic Alliance to develop this. We decided to partner with Genetic Alliance because they're not only using, have that relationship with Robert Wood Johnson, but they are also sharing insights and collaborating with FDA and with PCOR. And we knew that these were relationships that we wanted to establish and benefit from. And so additionally, the portal had been, had received some awards for being innovative. And so we felt that it was the right fit to partner with Genetic Alliance. So what data do we collect on the portal? We actually have a number of types of data that we collect. We collect demographics. So when you first sign up for the portal, we're gonna ask you about your bleeding disorder diagnosis and the severity. We're gonna ask if you're a parent, or a caregiver, if you're a patient, if you're a carrier. We're gonna ask you gender, age, demographics, and then we're also looking at including patient-reported outcome measures. So some participants have actually answered self-bleeding assessment tools and other types of patient-reported outcomes. And then we're also asking quality of life questions. So we want to know how is your general well-being, and how are you impacted by your bleeding disorder, and how do we manage your day-to-day life? How do you do that? And so we potentially asking about your activities of daily living, what are some other activities you engage in, your health, your outlook, and what does your support system look like? And finally, the portal also allows patients to share their medical records and even biological specimens. And so you can upload that type of data into the portal if you wish. And so who can use the portal? Anyone with a diagnosed bleeding disorder, whether it's hemophilia, VWD, a platelet disorder. We also encourage caregivers of children under 18 to join the portal, and any adult female who has either a child with a bleeding disorder or knows or believes that they're a carrier of a gene that causes hemophilia. And so it's important to us that all community members, whether they're directly or indirectly affected by a bleeding disorder, that they be part of the registry and the portal. So, will my data be safe? And the answer is yes. One of the things that we looked for when we signed up to be part of the portal through Genetic Alliance was that it had safeguards in place to protect the data. And so the type of security, the comparison that they continuously make, it's the kind of security that banks have. And so that's how safe the data is. It's protected. And also it's protected in the sense that only 2 of us at HFA have actually access to the data in the portal. So not even anybody else within our organization has access to that. And so it's really important to us to respect that and protect your data. So will HFA make my data available to non-HFA staff? No, not without your consent. And we are actually not able to grant access to anyone to the data portal. The data on the portal, only you can do that. And actually, only if you're satisfied, let's say we have a researcher come to us and wanting to ask access to the data to learn more about a particular topic, we ask you through the portal if it's okay to give a researcher access to your data. And we give you an opportunity to review the study that the researcher wants to conduct and how the data would be used by that researcher. If you're satisfied with that response, then you would say yes or no on the portal, and it's your choice, and you can always change your decision to share or not share your data. So what is new with HFA's portal? So you may have noticed that we have kind of been laying low a little bit for the past year and a half. We are actually completing a transition. Genetic Alliance has contracted a new provider for the platform. And so we're actually in the process of completing a transition to a new contractor, which is called Luna DNA. And so in the coming months, you're going to hear a little bit more about our new platform and what that's going to look like and how to transition your old account to the new account. And so we'll be, providing more information on that. And it will be available through HFA's website. And once you have an account in there, you will be able to choose surveys that you want to participate in and surveys that you don't want to participate in. And you'll actually also be able to use it in your mobile device, or whether you have an iPhone or an Android device or an iPad or some other tablet, it will also be available in a improved mobile format. So if you have any questions, feel free to reach out to us at research@hemophiliafed.org. We are more than happy to answer any questions that you may have, and we really hope that you'll take some time to check us out, check out the research portal, and let us know if we can answer any questions.
Well done, Mabel. That was great. So far, I'm just staying out of everyone's way. You speakers are really hitting upon so many important topics here and really doing a beautiful overview. I want to now pass the mic ever so gently over to Julie, who's last but certainly not least in this panel. Julie, can you just take a moment to tell us a little bit about yourself and about the patient notification system, and because in some respects you might be the most mysterious of all to our communities. I'm hoping that you can part the clouds and shed some light on things.
I'll do my best, Eric. Thank you.
Great.
And thank you so much to HFA for including me in this presentation this afternoon. I'm Julie Barkhofer, and I'm the Senior Vice President of Public Affairs for the Plasma Protein Therapeutics Association, PPTA. And as Eric said in his opening remarks, there's lots of acronyms in Washington, D.C. And we're a trade association. We represent manufacturers of plasma-derived and recombinant analog therapies, some of which are used by members of the bleeding disorders community. And PPTA, I'm going to talk about the system that we administer. We have a third-party, completely separate company run this system called the Patient Notification System. And the PNS is really in place to empower patients to give information to patients on product safety if there's a withdrawal or a recall. So we're not collecting data, we're not culling through or mining data. If you sign up, if you register, you will receive notifications. And that's what I'm here to talk to you about today. Next slide. So as I mentioned, we have a company named Sedgwick who administers the system. The system works when participating companies, manufacturers, they're the companies that make the blood clotting factors used by individuals with bleeding disorders, hemophilia, von Willebrand's. They notify the FDA if there's an issue, a safety issue with a product. And then Sedgwick, the administrator, to anyone who is registered for that therapeutic class or that product, the brand that you use, that you inject or infuse, you will receive direct to patient an automatic notification. And that's why we call it the patient notification system. It's open to all patients, physicians, family members, HTC staff, caregivers, any interested parties. You can go, and I'll give you the website at the end. It's not very creative. It's www.patientnotificationsystem.org, and you can register and you can be informed, because the manufacturers are committed to the communities, the patients that use the products that they make. And again, information is power. Next slide. So you may look at this list and you may hopefully see a company's name that's familiar to you. These are all the companies that are registered in the patient notification system that have their different brands of blood clotting factor therapies. Hopefully, as you look at this list, you see a company's name that's familiar to you. If you only know the name of the medicine that you infuse or inject, you could certainly ask your doctor, hey, who makes this brand or this medicine that I'm using? Next slide. And you can see here, PPTA, as I mentioned, I represent the industry, the manufacturers and the collectors of plasma, but we don't do this system alone. We work with other groups. And hopefully you can see in this list that Hemophilia Federation of America is clearly named. The Food and Drug Administration, the FDA, is part of our working group. And we get together and we talk about the PNS and we collaborate together on how can we improve the system so it is better for patients. Next slide. Now, I joined PPTA in 2001. And you can see here, this system was created in 1998. At that time, and think about 1998, think about technology 20 years ago, trying to get information out, trying to share important safety information about product recalls and withdrawals. It was a different era back then. Believe it or not, we had fax machines and some people actually selected fax as their preferred method of notification. It's a model system. It's been adopted in Canada, and therefore it's in both French and English. We, believe it or not, back in 2004, we included email. That was a big deal back then, that you could have your notification via email. And that is really good if you're traveling because it's easily accessible and it's instantaneous. The whole point of the PNS is we want you to receive the information before you infuse or inject your medicine, right? You want to know if there is a recall or a withdrawal. We call those an event. You want to know before you infuse. So we put email in place in 2004. We have Spanish brochures available, and that was really, I have to say, at the urging of the HFA, and we're grateful to them for always pushing us forward. We have a QR code. It's, you can just scan it with your phone, and it'll take you right to the website of the patient notification system, and you can receive your information. And just recently, we were able to add a new platform, text messaging. And you may say, oh my gosh, Julie, text messaging, that's so, that's been around so long, but it's very difficult to build things into a technology platform. And the companies that participate in the patient notification all equally shared the cost, so we could offer text messaging as an option for registrants, patients, physicians, family members, anyone that's interested to receive a notification. And again, you want to know before you use the medicine, the product, if it's safe. And that's what this is all about, putting patients and safety first and empowering patients with direct to patient notification. Next. Well, you can see here that there's been growth in the registrants of the system. And you can also see that there's about 9,700 total registrants. And if you think of all the people in the bleeding disorders community and other patient groups that used plasma-derived medicines, such as people with primary immune deficiency disorders or alpha-1, we only have 9,700 people. It's a completely free, completely confidential system, and it's just giving you direct information. HFA, as you'll see on the next slide, has been extremely influential. So take a look at the next slide in helping us increase the number of registrants. You can see here the vast number, over 50%, are patients. We have healthcare providers, that would be doctors and HTC staff. We have parents, that's very obvious, particularly in the bleeding disorders community, why parents are registered. And we have others. Others would be industry people, friends, family members, people that just really want to know or have an interest or some relationship with a patient who is using a plasma-derived recombinant or other type of factor treatment. Next slide. And you can see here the vast majority receive information via email. And only right now, that little yellow sliver, we really want to increase text messaging. We've worked over the past year to put out information that text messaging is available. We understand the utility of it is easier, and it's really obviously preferred by some of the younger members of communities. So we are working to increase the number of registrants that use text messaging, and I think we'll do that over time. Next slide. And this is where you'll really see the strength of the bleeding disorders community, and it reflects on the excellent job that the Hemophilia Federation of America has done to help us raise awareness in the community. The registrants that are under the blood clotting factor category are so much more prevalent than individuals with alpha-1, which is like a genetic emphysema, or immune globulin IG, that's an antibody replacement. For example, years ago, people heard of the story of the bubble boy. Immune globulin IG is an antibody replacement. And that's what these therapies do. They replace absent or missing proteins in people. But you can see here, the bleeding disorders community, you're doing your fair share, and we're very grateful and thankful to you for that, and to HFA. And then finally, the last slide is contact information. If you'd like to visit the website and register. No one at PPTA, I've been at PPTA 20 years. I've never even been to Indianapolis to the bunkers where they house these computers. No one has access to your records. It's not used for other marketing. It's not shared or sold. This is a complete system for patient empowerment and for patient information, for notification on the safety of your therapy before you infuse. And I thank you so much for your time. And Eric, I'll turn it back over to you and welcome any discussion. Thank you.
Thanks, Julie. That was great. In light of the heartbreaking history in our community, and even the last couple of years as a provider, we've gotten some important updates and alerts regarding certain products, your organization and the PNS is vital. And I think, again, like you said, and I should have said that at the top of the hour, but you can't say it enough. Information is power, and you got to stay involved, and you got to get you got to connect. So if I could ask Michelle and Mabel to come out of the shadows. There you are, and unmute yourself. We have just about 10 minutes left. Michelle, I do want to put you a little bit on the spot because you threw some great stuff out there, and I know Mabel spoke to it with regards to community accounts, the HFA research portal. Can you speak a little bit about how NHS in particular keeps participants' data safe? Because that's, I think, really important in our community. People, you know, as a provider, how important PHI is and how private it is. Can you reassure people kind of what measures y'all have in place over there?
Sure. And you were cutting in and out a little bit. So I understand that you're asking if what we do to keep data safe in the CVR portal is specifically. And our platform vendor is a vendor, it's called Arborimetrics, that is very specific to the healthcare institution. They do registries for hospitals, academia, and they have the highest level of patient HIPAA provider information security. And so they're constantly looking for any type of breaches. They have things that are very technical, you know, looking for what we call redundancies, all of that kind of stuff. And we chose them because of their expertise in that area, understanding, as you said, the history of our community and the fear of that. We did, even though the registry did not need to have IRB approval, we did go through IRB, and we actually have had IRB approval twice, where we put it to IRB and had them look at everything and have had their approval twice and have been exempted twice. And the agreement that we put forward to all of our participants is an agreement similar to what you would see in a clinical trial. It has all of the components of an informed consent because we want to be very transparent. And any information that comes out of CVR is reported in a de-identified and aggregate manner. So no individual person is ever identified. And we never put anything on the personal dashboard at a level that anybody could identify somebody else. And I think all of us understand how easy it is to identify somebody. It's like, oh, hey, I know that person in Michigan or Massachusetts who has that ultra rare disorder. So we never put anything on the personal dashboard that is at a the data is so low that there's a possibility that you could go, I know that person. I know who's answering that question. We filter everything out carefully.
Well done. Can y'all hear me okay? I feel awful that I was cutting in and out there.
No, it's not your fault. It's the internet where we're
technology. Man, I'm getting zoomed out. I don't know about y'all, but you threw out an acronym that I want to follow up on, IRB, the importance of protecting human subjects, the importance of ethics in research. Michelle, feel free to answer this, or Mabel, anyone can answer this, but can y'all elaborate on what IRB means and how that applies to research in our community?
Sure. And Mabel, do you want to answer it or do you want me to?
I'm happy to answer. You can jump in and add anything you want. So it's an institutional review board, and these are bodies that are usually established within different organizations or are contracted if they're standalone. And their objective is to review the project and review, the protocol, review the instruments or whatever activities are going to be done in a particular study, and then decide, if according to rules and regulations that are applicable, if this study provides enough protection and if people are going to be protected enough, and if there's any risks, if they're informed properly of the risks, and if they understand, what the consequences to participating in the study are. And so it's really a step in figuring out how do we ensure that the researchers are taking the proper precautions to protect the subjects.
And key to that is that the patient is always central. It's always looking at the person who is being studied or the participants of a study, and that their safety is always of utmost concern. And it's not just their physical safety, but also their psychological safety and their social safety also.
Absolutely. As much as this hour was devoted to registries, as a provider and employee at an HTC, we participate in clinical trials and research too. We all take IRBs to heart and want to protect our patients' rights as well.
Mm-hmm.
So thank you. I think you touched upon this at the top, Michelle. There's a lot of different registries, a lot of different research going on out there, and our community can get pretty overwhelmed. And do y'all have any advice on how our because as a social worker, patients and parents come to me. I don't know really what to sign up for. I don't know what to do. What advice would y'all give to our patients to kind of cope with this cornucopia of options?
Well, it is difficult, and it has to be based upon what their interest is. And I think that we are all different. Even the HFA portal versus CVR is different. And, CVR to ATHN and the HFA portal to ATHN is different. They all serve a different need. And if the person can find the time to participate in these different areas, of course, it's always valuable. But it comes down to what their personal individual sense of value is, and where they find that to be. I think that's really what is important, where they find the most value to them and where they want to expend their energy. And that's really, it's a personal preference for everybody. What would you say, Mabel?
Yeah, I agree. I think, one of the goals with this session is to lay out the options and allow people to understand what is out there. And so by doing that, you can understand what each of us is trying to do and what are our ultimate goals. And based on what aligns with your interests and your concerns and your preferences and what you'd like to see happen, you can sort of pick and choose, So one of the things that we're trying to deliver out of the session is sort of an infographic that, we can kind of see all of the options out there. And so look for that, as a outcome of the session. And then you can, take the time to think about it and really, talk to your family, talk to other community members, figure out what works for you. I think it's we're a community in a very real sense. And so how we can support each other in making these decisions is important too.
And the other thing is that some registries are very passive. The ATHN data set is very passive for a participant, because once you give consent, the treatment center is really doing most of the work. And then things like the HFA portal and CVR, they're more active and they're engaging you more often. But then they also offer more opportunities for you too. More opportunities for you to have your voice heard, and for you to be actively engaged. If you feel that research is important, and really thinking about the changes that have happened in the last, not even a decade, 5 years, to the landscape of treatment availability and the quality of life for our patients, that has come through research. And that has come because people in our community have stepped forward and said, yes, I will participate. I will give my information. I will be available. And that has made it easier for so many other people. So I think that's important for people to remember. In order to make life better for not only yourself, but your children and your families in the future, you really do need to step forward and make yourself available to these types of things.
Absolutely. You know, we're coming upon the 5 o'clock hour, and I do see that a gentleman named George put in his 2 cents and said, we're being surveyed to death. But I really appreciate Michelle and what you and Mabel said. Pick and choose. I mean, there's a lot of options out there. Just be, you know, like everything else in life, buyer beware. Just be an informed consumer and see what works best for you. Me, I'm a guy who likes food. When I go to a buffet, I don't eat everything along the food line. I ended up in the hospital. But you know, take a look at the menu, pick something that works for you, and the more people participate, the better it will be for our very small and intimate communities.
And I think George made another comment about not knowing what it's like to live with a bleeding disorder. And I think it's not necessarily to address the comment. It's not necessarily that we don't know and we don't understand, but I think it's that there's power in collecting data in a systematic way. And then to build the evidence in a way that it can support policy change and that it can support drug approvals. And so it's not just about the idea of whether or not we as individuals, you know, who are interested in research know what it's like and have had the opportunity to know community members' experiences, but it's really about how do we collect data in a way that it can be used to drive that change that Michelle was talking about.
Well said.
Exactly.
Well, on that note, I just do want to thank Michelle, Julie, Mabel for taking time. I really appreciate the all of our participants, all of our attendees sitting through this session. I want to be respectful to Dr. Clark and sign off on this session and wish you all a good weekend.
Thank you all.
Thank you so much to our panel. Really good information today, and we really appreciate you taking the time, especially on a Friday afternoon, to share more about registries and research that's going on in our bleeding disorders community. Next up, we have Dr. Dave Clark. Dr. Clark is a familiar face in our community. He has been a longtime volunteer for the Coalition for Hemophilia B, has been a staple of the On the Horizon session at the HFA symposium. Dr. Clark is a chemical engineer by profession, but is very passionate for our community. And we're so honored today to have you talk with us more about the emerging field, what is coming down the pipe in bleeding disorders. So Dr. Clark, I'll turn the floor over to you.
Hi, everybody. Thank you for that introduction. And today I'm going to talk about emerging therapies, sorry. You've spent the last couple of days hearing about a lot of the products that are already on the market. And today I'm going to talk about some that are going to be coming along in the future. Some of the things I'm going to talk about may eventually make it to the market. Some of them may not, but that's just the nature of research and product development. So if I could have the next slide, please. Disclosures, I have nothing to disclose. I'm retired and I don't work for anybody. I'm a volunteer. I'm actually, as a volunteer, I'm the chairman of the Coalition for Hemophilia B. And let me just put in a little blatant ad here for the coalition that if you're watching and you have hemophilia B and you don't know about us, I would encourage you to learn about us. You can go to our website, which is hemob.org, and see what we're all about. We have a lot of good programs that help support the B community. So the next slide, please. Okay, here's a chart that just kind of shows the evolution of hemophilia treatment over the years. I started as a pharmaceutical scientist. I was still a little ways along in my career, but I first started working with hemophilia in 1982. So you see, I started pretty far back on the timeline there. And at that point, we had fairly good concentrates of factor VIII for hemophilia A. Hemophilia B was a different story. The concentrates there were not very pure. Turns out it's difficult to purify factor VIII away from some of the other clotting factors. So the factor VIII concentrates that were developed then couldn't be used in large doses or for long periods of time. So they couldn't be used. They were fairly dangerous to use in surgery and couldn't be used at all for prophylaxis. But we've come a long way since then, as you see there with all the new products that have been developed over the year. And we're up to the end there in 2021, and I'm going to be talking mostly about the products that are listed in the gray box, all the way down to gene therapy, which might actually at some point become a cure for hemophilia. So if I could have the next slide, please. All right, first we're going to talk about factor replacement therapies that are in development. Factor replacement therapies are the products that you're already using, either factor VIII or factor IX that you infuse by IV infusion to replace the defective factors that your body makes because you have mutated genes of either factor VIII or factor IX. Let's go on to the next, please. First, let's talk about product development. As I say, I started out in 1982 in product development. So I've been through this a lot with all kinds of different products and a lot of different clotting factor products. But we start out at the R&D stage, research and development. And this is mostly product discovery. And then when you think you got a product, you have process development so you can figure out how to make it. And this is a very, in a way, very tricky part. You're just playing around to see if what you've got really might work and might be a product that could be useful for people. And then when you decide that, you go into preclinical studies. And these are more formal studies, some done in the lab, some done in animals, to just make sure that the product you have really does look like it's going to work, and most importantly, that looks like it's going to be safe. So we do toxicity studies. One of the important things that people are mostly not aware of is that we do stability studies. We have to make sure that this product that we're developing is going to last. Most products are dated for about 2 years. You might have the best product in the world, but if it only lasts for a couple months in the vial, that's not something that's really useful. We saw some of this actually with the COVID vaccines, where the Pfizer and Moderna vaccines originally had to be stored at -80 degrees centigrade, which was a big problem. Now they've relaxed that a little bit, but you've got to make sure that your product, that you really have a product that's going to be stable and safe. So then when we've gone through the preclinical studies, at the end of that, you present all your data to the FDA in a document called an IND. IND just stands for Investigational New Development. And so the first thing you do is go into a phase 1 study where you give the product often in smaller doses, and you give it to a small number of volunteers just to make sure that the product really is safe in humans. Sometimes it looks like it's safe in animals, but then when you get to humans, you find there are new problems that you hadn't seen before. So you go through phase 1 to just make sure that it's really going to be safe in people. Then you go into phase 2, which is dose determination. In phase 1, you usually start with just very small doses. You just want to build up gradually to really make sure your product is safe. But when you get into phase 2, you want to start figuring out what dose of your product people really need to have the effect that you want. And when you've done that, then you go into phase 3, which is a trial in a large population. Now, if we were talking about cancer or heart disease, a large population would be thousands of people. We don't have thousands of people to do this in. So usually for phase 3 studies, a large population in hemophilia is more like 100 or so people. And then so the phase 3 studies are really what you use to provide data for your license application to the FDA in order to get your product licensed and be able to put it out on the market. So the phase 3 studies are really the crucial studies for whether your product is going to be something that the FDA will approve and something that you can market. For some products, you have to go through a phase 4 study, which is a post-licensure study. The FDA may have enough confidence in your product to give it a license, but they still may have some questions. So they may ask you to do a phase 4 study to study more patients or patients under some different conditions, just to make sure. We don't see that many products that go through phase 4 studies, although I think probably gene therapy, they will have phase 4 studies because that's something we're expecting to last for a long time and really have to watch it for a while. The other reason you might do a phase 4 study is, for instance, to get a new indication. Your product may initially have been approved just for use in adults. You might want to do a phase 4 study in younger people to get an indication for its use in kids. So this is basically what, there's a lot to each of these, but that's just a little nutshell of what each one is. Let's go on to the next slide, please. Okay, first we're going to talk about half-lives. The big thing going on in hemophilia treatment, as you know, is trying to get products that have extended half-lives so that they last longer in circulation. The original idea for extended half-lives was so that you wouldn't have to infuse as often. Instead of infusing several times a week, maybe you only have to infuse once a week, or for some of the factor IX products, even once every 2 weeks. And in factor IX, we've made some good strides in that. Let's look at the factor IX column first. You see plasma-derived factor IX has half-lives up to about 25 hours. And the same thing with recombinant factor IX products, again, up to about 25 hours. There's a whole range in each of these, not so much because of variations in the product, but more variations in the patients. One of the important principles in medicine that people don't talk about enough is the fact that everybody is different. For any drug or any treatment, if you get 100 people, you're going to have 100 different reactions to the product. It may work better in some people than in others. It may not work at all in some people. You just don't know till you really try it. But it's the same thing with half-lives, is that some people seem to have longer half-lives for some of these products, and some people seem to have shorter half-lives. So then we go to the extended half-life factor IX products. And you can see we've made a big jump there from about 25 hours up to 83 to 104 hours. And that has really made a major difference for a lot of the hemophilia B patients. because as I said, now they can infuse maybe just once a week, or for some of them, even once every 2 weeks, or even a little more. So let's get back to the factor VIII column. And there you see plasma-derived and recombinant are again about the same, with maximums around 17 hours. But then we go to the extended half-life factor VIII products, and you see there's not much of a difference. We've only gone up to about 19.7, maybe 20 hours. And why is that? These extended half-life factor VIII products use the same methods that the factor IX products use to extend the half-life. And yet they work very well for factor IX, but not much at all for factor VIII. The reason for that is in the third column, the von Willebrand factor. You can see that it has a maximum half-life, again, around 20 hours. What happens with factor VIII is that it doesn't circulate around in the bloodstream by itself. It binds to von Willebrand factor. Factor VIII by itself is a big molecule, and it's really not very stable. In fact, when we're making factor VIII in production, we have to work quickly because factor VIII tends to fall apart just by itself. So it binds to von Willebrand factor, which tends to stabilize the molecule. But the problem is, while it's circulating around with the von Willebrand factor, it's really the von Willebrand factor's half-life that controls this. So to some extent, no matter what you do with factor VIII, it's dependent on the half-life of the von Willebrand factor. And I'm going to, in the next couple of slides, I'm going to show you some ways that we get around that. But and then the final row in the table there is Hemophilia, and it's hard to give it an actual half-life, but it has a half-life of about 4 weeks, which those of you that are on Hemophilia know is a real benefit. You don't have to do this every other day infusion or anything. So let's go on to the next slide, please. Okay, here's an extended half-life factor VIII product that's under development. It's called BIV001 from Sanofi. It's in phase 3 studies, and it gets around this problem of binding to von Willebrand factor and having a shorter half-life because of that. Let's go to the next slide, please. At the top of this chart, you see a little drawing of what BIV001 looks like. The A1, A2, A3, and C1, C2, that part together is B domain deleted factor VIII. Factor VIII is even larger than that. It's got a large B domain, but it appears that the B domain is not really involved in clotting. So most of the time nowadays, we work with B domain deleted factor VIII, which seems to work just about the same as normal factor VIII that's in circulation. Now you see the two little FC globs to the right of that molecule. If you take the A1, A2, A3, and the C1, C2, and those two FCs, that's eloctate, one of the extended factor VIII products on the market. This FC region, the FC is a part of an antibody molecule. An antibody molecule is shaped like a Y, and the two arms are what binds to whatever the antibody is directed against. If it's a virus, then the two arms bind to the virus. If it's a foreign protein, they bind to the foreign protein. What happens, and then the base of the Y, the up and down part of it, is the FC region, and C stands for constant. That's the part of the antibody molecule that is the same in all the different antibodies. And it turns out that the body has a special system to keep antibody molecules in circulation longer than most other proteins that are in circulation. And so by hooking the factor VIII molecule onto this, we take advantage of that. And the FC region, when the body goes to, well, in the body, the body is constantly making new proteins and getting rid of old proteins. And that's mostly just to keep all the proteins that are in circulation in good shape. They tend to get banged up a little bit in circulating around in the bloodstream. So the FC region, there's a system that when an antibody molecule comes in to be broken down, degraded, the system recognizes the FC region and says, no, this is an antibody. Let's wait a minute and keep it in circulation a lot longer because we might need it. There might be an infection that needs this antibody. So it works the same way with the factor VIII, that the FC region will keep the factor VIII molecule in circulation longer. But still, because the factor VIII is bound to von Willebrand factor, it doesn't really happen that well with factor VIII. It works very well for factor IX, where we get up to like 100 hours half-life. But at the bottom of that little drawing, you see a D prime D3. That's a little chunk of von Willebrand factor. That bottom of the molecule there is where the factor VIII binds to the von Willebrand factor molecule. Well, instead of that, we've taken the little chunk of von Willebrand factor that the factor VIII binds to and stuck it on there, and that helps to stabilize the factor VIII molecule, but it also keeps von Willebrand factor from binding to this factor VIII molecule. So with that, this part of the FC portion there will tend to keep the factor VIII in circulation longer. So just by preventing it from binding to von Willebrand factor, we can really increase the half-life. And then you see there's a couple little blue-gray squiggles there. That's a long chain of amino acids. Proteins are made of amino acids. Proteins are long chains of amino acids that are in a specific order and with a specific kind of structure. I'll show you a little bit of that later. And so these are little pieces of protein, except they're amino acids that don't have any structure, so that it just is a long chain of amino acids that kind of is loose and moves around, and kind of it kind of wraps it. They kind of wrap themselves around the factor VIII molecule and keep it from being seen as well by the cells that want to break down the molecule. This is the same kind of way that the PEG works. Any of you who are on Rebalancing for factor IX or on GV or Esproct for factor VIII, those have polyethylene glycol chains on them, which is very similar to this. And those PEG chains kind of wrap around the molecule and kind of hide it from the parts of the body that want to degrade it and take it out of circulation. So on the graph down below there, you can see that this actually works, that with recombinant factor VIII, we have a half-life of about 7.5 hours. With recombinant factor VIII FC, which is the lactate, that raises to a half-life of about 16.5 hours. But with the BIV001, with all these extras added onto it, we can increase the half-life to about 32 hours. So, for those of you that are just on regular factor IX product, or factor VIII product, sorry, this is a new product that's going to be coming along that should give you a real extension in the half-life of the product and really cut down on how often you have to infuse. Now I say, maybe is because we never know with these products whether they're going to make it through all the clinical studies and finally get licensed. But this one seems to be coming along pretty well, and I think there's a good chance it's going to be on the market in a few years. So let's go on to the next slide, please. Okay, so these are more extended half-life factor 9 products that are being developed. One of the big things we've realized lately is that if you inject these products subcutaneously, that's what the SQ means under the administration route. If you inject them sub-Q, that by itself kind of gives them a longer lifetime in the body. If you, because basically you have a little reservoir of this product under the skin that just slowly diffuses into the bloodstream. So, basically you can call on that little reservoir for a few more days, and that just by itself, just the administration route, not only is an easier administration route than intravenous, but that is administration route actually gives you a longer effective half-life. Actually, this is not a new concept. Back in the 1980s, when I was working for the American Red Cross doing research on plasma proteins, we tried to develop a subcutaneous factor 9. The problem is that your skin is your skin is a barrier between you and the rest of the world. And in a sense, it's really part of your immune system. Your skin is there to protect you against whatever might be outside of your body. And because of that, your skin contains a lot of what are called proteolytic enzymes. These are enzymes that break down proteins. They break down things like factor 8 and factor 9. So you can't just take anything and squirt it under your skin subcutaneously and expect it to work. You've got to really protect it while it's there under your skin, because your skin thinks it's something that shouldn't be there and wants to get rid of it. So a big part of this is just finding ways to formulate the products so that they're stable when injected underneath the skin. So at the top one there is dalsanonicogue alpha, which we call DALK-A for short. We're going to start seeing more and more of these long names. And these are the sort of international generic names, and there's a whole system of how to name these. The nonacogue, you see cog refers to coagulation or clotting. Nona is for 9 or factor 9. And then the first part of the name, the manufacturer can just pick, and they can pick anything they want. Those of you who are on Hemlibra, it's an interesting story. Hemlibra, its generic name is emicizumab. Well, sizumab is the part that's required by the international naming system, but the manufacturer can put anything they want in the front. So emicizumab was first developed by Chugai, a Japanese company, and the Japanese tend to be kind of playful with these things. So Emi means smile. So if you're on Hemlibra, you're supposed to smile. So, and then we see BIV002. This is another factor 9. The first part of the name, RFIXFC, that's the same as Alprolix. And then they're going to put this X10, these little chains of amino acids on it to try to give it a longer half-life. And that will also help protect it as a subcutaneous injection. And then CL cell bearing, we don't know much about this yet, but they're doing the same thing with their Idelvion, as they're trying to make a subcutaneous version of it. So this is, you can see here, this is where we're going with extended half-life factor 9 products. So next slide, please. Okay, here's the dalsanonicogue alpha or DALK-A being developed by Catalyst Biosciences. And so the main drawing there is kind of a schematic of the factor IX molecule. This is what I was talking about protein structure, is that you can see the parts that are just single lines, that they're curvy lines. That's just, those are just the chains of amino acids that make up the protein. In this case, factor IX has 416 amino acids strung together. But then you see other shapes. Some are kind of like spirals and some are kind of flat sheets. And those are structures that are important for the function of the protein. In the spirals, the amino acids tend to form a spiral like that and make a more stable little piece. And the same thing with the flat sheets. In the flat sheets, the chain is actually zigzagging back and forth across the sheet. And that's important for the function of the protein. If you were to take this protein and boil it and just totally denature it, so it was just one long string, you wouldn't know if it was factor VIII or factor IX or anything. It would just be a long string of amino acids that wouldn't really do anything. So it's the structure of the protein is really important. Now, what Catalyst did with factor IX here is they had a method of making thousands and thousands of small variations in the factor IX molecule. And then they had a method to screen these rapidly to find the ones that look like they might have better factor IX activity or a longer half-life, or preferably both. And so this is one of the molecules that they selected out. And you can see that it really only has 3 different changes in the amino acid structure. The R318Y there gives it a greater resistance to antithrombin. Antithrombin is an anticoagulant. It's part of the control system in the clotting system, and it tends to inhibit the activity of factor IX. And then you see the other 2 down at the bottom there give it, that actually should say factor IX, not factor VIII activity, but it gives you, oh no, I'm sorry, that's right. It's increased factor VIII affinity, so it sticks to factor VIII better. Factor VIII and factor IX work together in the clotting system, which I'll show you in a minute. So that's the reason that hemophilia A and hemophilia B are not identical, but they have very similar aspects, because it's really this factor VIII, factor IX step that they inhibit, so that if you're missing either factor VIII or factor IX, or have defective versions of those, the clotting process kind of stops right at that step, whether it's factor VIII or factor IX. So the one thing I want to point out here is how just very small changes can have a big difference in how these proteins work. And this is partly what genetic engineering is all about, is trying to find out how to put in these small changes and get them to make big changes in the performance of the molecules. So let's go on to the next slide, please. This is the BIV002, again from Sanofi, and this just shows how it's, what it kind of looks like. On the left-hand side there, you have a normal recombinant factor IX. This is equivalent to Benefix. The WT up there, it says WT catalytic. WT stands for wild type, which just means normal factor IX. This is how you find factor IX in nature, in plasma. And catalytic is that factor IX is an enzyme that catalyzes reactions. It specifically catalyzes the activation of factor X. So then we go to Alprolix, which was an extended factor IX, extended half-life factor IX. And you can see there they've just stuck on these FC molecules, and that gives it a much longer half-life, just because the body then looks at it as though it's an antibody that should stay in circulation longer. And then the next step there is actually a couple different things. First, they're changing from the wild type factor 9 to the Padua variant of factor 9. This is an interesting thing that they found a family in Italy that instead of having, they had mutated factor 9, but instead of having less factor 9 activity, this mutation actually gives their factor 9 a lot greater activity, to the point where actually the family suffers from thrombosis, which is too much clotting, and that can be very dangerous. But the industry has largely, and this Padua factor 9 just varies from normal factor 9 by 1 amino acid. So the industry's kind of jumped on this and said, wow, here's a way we can have a much more potent factor 9 product by using this Padua variant. And then they also stick on this X10 little loop. It's actually longer than it looks here, but it kind of wraps around the molecule and gives it even more half-life because it kind of hides it from circulation. So let's go on to the next slide, please. Okay, now those of you who have inhibitors know that that's a big problem. And the 3 inhibitor treatment products that are on the market now, Pfiba and Novo7 and 7-Fact. Novo7 and 7-Fact are both activated versions of factor 7 that get clotting started by a slightly different mechanism. I'll show you that a little bit later. Pfiba is a whole mixture of activated clotting factors, but probably also includes factor 7. So those are basically the products that you have to work with now. And you know that if you have inhibitors, that they don't work very well. They have very short half-lives. If you're really having a bad bleed, you might have to infuse several times a day even. So there's a big push to try to find better factor 7A products. These are called bypassing agents because they bypass the need for factor 8 or factor 9. So again, Catalyst has come up with this Marzepticog alpha or MARZ-AA that again is one of these variants that they found that has a longer half-life and a greater activity. And they're in phase 3 studies with that, and it seems to be performing pretty well. It's also a subcutaneous injection. And so this is a product that may make it on the market in the next few years. The next one down, Opco Health, they're looking at theirs. This is another factor 7A product that has a longer half-life. And they're looking at both as an IV and a sub-Q injection. So for IV injection, they're in phase 2A, and for sub-Q, they're just starting out with phase 1. And then Novo and Sanofi are also working on improved factor 7A products. We don't know that much right now about Novo's product. Sanofi's product is again a product that has this X10 amino acid loop on it to give it a longer half-life. So let me go to the next slide, please. Factor 10. Now this is an interesting thing. And a lot of people are working on it, although you see that just about everybody is just in R&D studies, just research studies in the lab, except for Pfizer, who started phase 1 and then gave up. Factor 10 is, I'll show you on the, I have a schematic of the whole clotting coagulation system coming up. But factor 10 is where it's kind of the central factor in coagulation, that there are 2 pathways, and both of them end up with activating factor 10. And then activated factor 10 goes on to to eventually activate some other proteins to form the clot. So the idea is kind of like the same thing with inhibitor patients for factor 7A, is that you could give people activated factor 10 products and not have to worry about factor 8 or factor 9 activating them if you don't have good versions of factor 8 or factor 9 in your bloodstream. The problem with factor 10 is it's really too good in terms of clotting. It's basically described as toxic, that if you just give somebody a bunch of factor 10, they're probably going to have a lot of thrombosis. And that was probably the problem with the early factor 9 concentrates, is that they contained a lot of factor 10. And nobody knows exactly why why those products caused so much thrombosis. But at the Red Cross, we thought one of the big reasons was that they contained a lot of factor 10, and people would basically get an overload of factor 10. So you have to work with factor 10 very carefully, because you can really tip the scales over to getting too much thrombosis, too much clotting instead of not enough. So you can see Catalyst there again has another variant, and Children's Hospital of Philadelphia, they do a lot of hemophilia research, and they've got another variant. CSL Behring is working on a factor 10 that gets activated by factor 11. Normally, factor 11 activates factor 9, and then factor 8 and factor 9 together activate factor 10. Well, this kind of drops factor 8 and factor 9 out of the equation so that factor 10 gets activated directly by factor 11. Novo is working on a thrombin activatable factor 10. And again, this is a way to keep factor 10 under control. You can't just give people activated factor 10, but there's a lot of thrombin that's generated in clotting. And if we wait and use that thrombin to activate factor 10, that may work. And then Acta10 from LFB, we don't know too much about it yet, but that's another factor 10. Now, whether these factor 10 products are going to go anywhere or not is hard to say. You can see that most of them, except for the one that got discontinued from Pfizer, are just in the research stage. But this is potentially another way around the not having enough factor 8 or factor 9. This would be you could use this for inhibitor patients, or you could potentially use it for just non-inhibitor hemophilia patients. And we'll just have to see where these go. So let's go on to the next slide, please. So now we're going to talk about non-factor replacement therapies. So as I said before, replacement therapy is basically giving you injections of the factor 8 or factor 9 that your body needs in order to clot. Well, these non-factor ones are saying, instead of giving you factor, we can give you something else that kind of will do the same job. And in fact, this is exactly, the first non-factor product on the market is Hemlibra, which is what's called a bispecific antibody. The job of factor 8 is basically to bring activated factor 9 together with factor 10 so that the factor 9 can activate the factor 10. Factor 9 is an enzyme that causes a reaction, and factor 8 is what's called a cofactor, which greatly increases the activity of the enzyme. So you need both factor 8 and factor 9 together in that step of the clotting reaction. And Hemlibra gets around that by kind of mimicking factor 8, that it's what's called a bispecific antibody. I told you antibodies are shaped like a Y, and it's up at the top tips of the arms that they bind to whatever they're going to bind to. Hemlibra is what's called a bispecific antibody because each of the arms binds to something different. One of the arms for Hemlibra will bind to factor 8, and will bind to factor 9, and the other arm will bind to factor 10 and bring them together just like factor 8 would normally do, so that the factor 9 can activate factor 10. So that's Hemlibra is the first non-factor replacement therapy, and there's, as you'll see, there's a whole bunch of other ones being developed that work by all kinds of different mechanisms. So let's go on to the next slide, please. Okay, here are two more products that are going to work kind of like Hemlibra does. Hemlibra has been, as many of you know, has been a big success. And so the other companies want a little piece of that action. So here's two others, one by Novo Nordisk and one by Roche Genzyme, that are basically going to be second generation versions of Hemlibra. And basically improved versions of Hemlibra. So you can see both of them are pretty much up to the phase 2 stage. So that's something come along. Hemlibra is potentially going to have some competition. Let's go on to the next slide, please. Okay, rebalancing the coagulation cascade. This principle has had a lot of different names, and I've had some different names I've used for it in my talks, but the name that seems to be coming out in the most use is rebalancing the coagulation cascade. Can we go to the next slide, please? So these new products will be called rebalancing agents, unless somebody comes up with a different name. This is a kind of a drawing of the coagulation cascade, the clotting system. And I'm not showing you this to show you anything except that it's extremely complex. And in fact, this is just a simplified version of kind of an approximation. It's actually much more complex than this. And there are parts of it that we still don't really understand how they work. But this is kind of, my reason showing you this is just to see that it's very complicated and involves a lot of different proteins. Now, two things are important about that. One is that it contains a lot of redundancy. If one part of it turns out to be a little bit weak, then it can actually compensate for some of that. And this is kind of the what's behind mild and moderate hemophilia, is that you may have a factor 8 or factor 9 molecule that isn't really, it still kind of does the job, but isn't really what it's supposed to be. And in most cases, the clotting system can kind of make up for that with other pathways. You can see there's all kinds of loops and things here. That there's all kinds of alternative pathways. There's two main pathways, but then there are a lot of other ways that it can compensate. So what happens when you have hemophilia and you have bad versions of either factor 8 or factor 9, is that the system here gets out of balance. Normally, there's a balance here between clotting factors and anticoagulants. There are a lot of anticoagulants in this system here that are basically the controls on the clotting system. They keep it from causing too much clotting or causing thrombosis, and keep everything localized at the site of the injury, so that you're not clotting all throughout your body, which, as you can imagine, is not a good idea. So what happens when you don't have factor 8 or factor 9 is that things get out of balance, so that you have a much higher level of anticoagulant activity than clotting activity. So the idea of a lot of these products that are being developed is to kind of rebalance things. So basically, they're trying to inhibit some of the anticoagulants to bring things back more in balance, so that things clot when they should and don't clot when they shouldn't. That it sounds simple, but it's not. They've actually had a lot of success with some of these, but there have been problems that have come up along the way. And actually, while I'm on this this slide, let me just show you quickly one of the things I was talking about before. Is what this kind of simulates is the inside of a blood vessel, and you can see at the top there's an injury where the blood vessel is broken apart, and there are two arrows going down out of that. One goes to this lighter red section in the middle that's called the intrinsic pathway, or you can also call it the tissue factor pathway, or I like to call it the factor 7 pathway. And that pathway is fairly simple, and that's to get clotting started right away whenever you have an injury. So it's a simple pathway just to give you kind of a little shot of clotting activity until the other pathway gets going. And you can see that it filters down through a few proteins there and ends up down converting factor 10 to factor 10A, to activated factor 10. The other one is much more complicated on the left, the darker red pathway. That does a lot of different things. There's a lot of control built into that. It also amplifies the signal. So basically the signal when you have a broken blood vessel is that there are two proteins that the blood then sees, tissue factor and collagen, and those get things started. But there's really not enough of a signal there, or not enough of the tissue factor or collagen in a lot of cases to give you as much clotting as you need. So this left-hand pathway that includes factor 8 and factor 9 down near the bottom kind of amplifies the signal and keeps it going so that you get enough clotting and as much clotting as you need. But you can see that it also comes out at the bottom there, activating factor 10 to factor 10A. So this is what I was talking about earlier, that activation of factor 10 or activated factor 10 is kind of the centerpiece in this. And then on the right-hand side, you see where factor 10A converts prothrombin to thrombin, and thrombin converts fibrinogen to fibrin, and fibrin is what forms the clot. But you can see also there's a lot of other feedback loops and things there to keep you from having too much clotting. So let's go on to the next slide, please. Okay, one of the anticoagulants that a lot of people are working on is called tissue factor pathway inhibitor. That's an anticoagulant that tends to inhibit coagulation or inhibit clotting. What we're actually producing is we're producing molecules that can inhibit the inhibitor and basically get it out of the way to let clotting go on more easily. So you can see that there are at least 5 different companies working on these things, and they have different methods and different kinds of ways they approach this. You can see that in the middle row there that Bayer discontinued the project because of thrombosis. That's the big problem in this, is that if you start inhibiting anticoagulants, you're basically taking away some of the control of the clotting system. A lot of people describe the anticoagulants as kind of the brakes for the clotting system. So if you get rid of some of those brakes, you can easily get us into a situation where you end up with thrombosis, too much clotting. Where this especially comes in, if you just take these, so far everybody has seen safe results and no thrombosis. The problem is if you have a breakthrough bleed, and you need to give somebody, for an inhibitor patient, either giving them factor 7A, or for a non-inhibitor patient, giving them factor 8 or factor 9. you've got to control those doses very carefully, because if you give them too much, you can easily send them over, basically tip the balance over towards thrombosis, towards too much clotting. And most of the people that have worked on these kinds of things have had problems just with this. You can see Novo Nordisk's concizumab. They just last fall had some problems with thrombosis, and the FDA put a hold on their clinical trial until they could get that figured out. So they basically come back and put more stringent requirements on just how much factor 8 or factor 9 or bypassing agent that you can give somebody if they have a breakthrough bleed. Pfizer so far hasn't had any problems, but you know, we'll see whether they do or not in the future. And then the two at the bottom from GC Pharma, which is a Korean company, South Korean company, and Takeda are pretty early on, so we don't really know exactly what's going on with them yet. So let's go to the next slide, please. Okay, now tissue factor pathway inhibitor is not the only anticoagulant. And in fact, I'm not really sure why exactly they kind of all focused on that. But these are some other products that target different anticoagulants. Fitusiran is at the top there from Sanofi. It targets antithrombin, which is another kind of general anticoagulant in the system. They're in phase 3 studies, and they've been actually coasting along very well until recently. And they've had some problems with patients who had breakthrough bleeds and developed signs of thrombosis. The way that fitusiran works is it's actually a molecule called an RNA inhibitor. It actually binds to when a cell in the body, in this case, pretty much the liver cells, when they want to make a protein, they take the gene and make a copy of it, not in DNA, which the gene is, but in RNA. And that RNA goes out into the main body of the cell, where it's translated into a protein like antithrombin. Well, fitusiran and also Silence Therapeutics, anti-protein S at the bottom there, both are working using RNA interference, where they basically bind something to that RNA so the body can't make that protein. Fitusiran, Sanofi really just recently realized with this that they're probably getting their antithrombin down to too low a level, and that they actually needed to leave a little bit higher level there just to try to maintain more control of the clotting system. So they basically they had a hold for a while, but now they're working again with different dosing guidelines. The two in the middle there, Serpin PC and HAPC 1573, excuse me, both target activated protein C, which is another anticoagulant. The nice thing about targeting that is that protein C by itself doesn't do anything until it's activated. So if these products only work on activated protein C, the protein C only gets activated at the site of injury. And so this will kind of localize it more and is less likely to cause thrombosis. That's the message theoretically. We'll see how that works in practice in the clinical studies. And then another anticoagulant called protein S at the bottom, Novo and Silence Therapeutics are trying to inhibit protein S. So, you know, we'll see how these go along. This particular area has a lot of potential, I think, but as you can see, everybody's been having problems. And so there's a lot of, you got to be more careful, I think, than people thought in the beginning to try to keep this all under control. Let's go on to the next slide, please. Here's a couple of other interesting products that don't really fit in any of the other categories. A company called Tremo is working on a product called Rofecoxib. Rofecoxib was initially on the market as Vioxx, and some of you older people may have used it for joint pain. It apparently works very well for that. Everybody I talked to is really sorry that Vioxx was taken off the market. Vioxx basically was such a good product that people just overused it, and that seemed to lead to heart trouble. But Tremo seems to think that they can bring it back in, under more controlled conditions in hemophilia, where people are not just going to be, it's just for a specific type of pain, and people aren't going to be taking handfuls of it because they're in so much pain from other things. It's mostly just going to be joint pain. So they're already in phase 3 studies because Vioxx actually showed already that the product works. And so they just have to show it for this hemophilia arthropathy indication or hemophilic joint damage. Clot Chip is another interesting little product. They're developing a portable clotting analyzer. When you go to the HTC or to your doctor and they take a blood sample, they have to send it away to get it analyzed to find out how much factor 8 or factor 9 you have in your bloodstream. Well, this would be a little portable clotting analyzer that the doctor could just have in his office and just take a blood sample and give you your factor levels right there. And actually, I don't have it on this chart, but there's another company working on something similar to this Clot Chip. So it's possible that we may have more kind of real-time monitoring of our factor levels. Let's go to the next slide, please. All right, gene therapy. This has a lot of potential. I'm sure you've heard a lot about it already. It probably is eventually going to be the cure for hemophilia. I don't know that the first products coming out necessarily are going to be, although they're probably going to be very good products. Most of them are essentially converting severe hemophilia into mild or moderate hemophilia. And so that makes a big improvement, but as we've seen in a number of studies over the last few years, even people with mild and moderate hemophilia can have a lot of problems, and they have different problems than people with severe hemophilia. So it's not a panacea yet, but it's certainly a step along the way. Let's go to the next slide, please. Yeah, so what is gene therapy? Gene therapy is either the introduction or alteration of genetic material, basically, which is made out of DNA, within a cell or organism with the intention of curing or treating a disease. So basically, you've got defective factor 8 or factor 9 running around in your bloodstream. What this does is put a new gene into your liver so that it can produce copies of normal factor 8 or factor 9 proteins. And by doing that, restore the clotting ability of your blood. So far, the gene therapies that are in clinical trials all use basically the same kind of approach, but with different variations. They use adeno-associated virus, or AAV, to introduce a new gene for either factor 8 or factor 9 into the cells of your liver. Let me just stop here for a minute and say AAV, adeno-associated virus, is actually, even though the name is similar, it's very different than an adenovirus. A lot of people have been concerned because some of the COVID vaccines use adenoviruses, and they're worried that they might become resistant to that virus and then not be able to have gene therapy. But that's not true. The AAV is not the same as a normal adenovirus. So let's go to the next slide, please. So basically, they're using a neutralized AAV to deliver a functional version of the gene or the DNA for factor 8 or factor 9. By neutralized, I mean that the viral genes are removed, so it's no longer an infectious virus in the sense that it's not going to reproduce itself or cause disease. Instead of the viral genes, they put in genes for factor 8 or factor 9, and so that little virus then with the factor 8 or factor 9 genes in it is called a viral vector. So then they take huge amounts of these viral vectors and infuse them into the patient. And since AAV normally infects the liver, they basically infect liver cells with this AAV. But instead of causing disease, they cause production of factor 8 or factor 9. Once the AAV gets into a liver cell, they kind of unpack themselves and stick the genes that they're carrying into the nucleus of the cell, which gives the cell basically the gene to produce the clotting factor, factor 8 or factor 9. So it's really as simple as that, but it sounds simple, but it's not, there's a lot of little problems along the way. It's not as simple as we thought it would be in the beginning 20 or 30 years ago. One of the things I like to point out is that NHF had a slogan back in the 1990s that they would have a cure by the end of the century. Well, that didn't quite work out, but we're still on the road to that cure, I think. So if I could have the next slide, please. Here are all the hemophilia A gene therapies that are currently in clinical trials. And the two that are farthest along are at the top there. Here's another, for BioMarin, there's another long name. But actually, for short, they're calling it Valrox, which is the first part of each of the two words in the name there. It contains a gene for B-domain deleted factor 8. As I said, we've realized that the B-domain, it probably has some other function in the body, but it's not needed for clotting. And they're in phase 3 studies. In fact, last summer, they submitted a license application and thought they were going to get licensed and be on the market by now. It turned out that the FDA still had some questions. The big thing about these are intended to be long-term products that you get one injection, but then it basically treats you for a long period of time, theoretically for the rest of your life, but we don't know. Because this has only been going on for a while, we don't really know if that's going to be true or not. And then you see Spark Therapeutics has a similar one that's also in phase 3 studies, and they're also getting good results. One of the things we're seeing in the hemophilia A gene therapies that we're not seeing in the hemophilia B gene therapies is that the factor 8 production doesn't last as long. BioMarin thinks their Valrox may last for like 8 or 9 years, and then you'd have to have gene therapy again. They see declining production of factor 8 over time. And some of the others have had similar indications that the factor 8 or factor 9 that the factor 8 production might be kind of dying out after a number of years. So this is one of the things that the FDA wanted. They want to follow these patients for longer periods of time before they get licensed. Nobody really knows why this happens, although we're starting to get some hints. A lot of this has to do with the immune system. And in spite of how bad the pandemic has been, one of the things that the COVID pandemic has really helped is research in immunology. We know much, much more about the immune system than we did back at the beginning of the pandemic. And I think that's only going to help us, not just for things like COVID, but for a lot of different things like gene therapy as we go along. You see the one at the bottom there from Takeda, and you see it's got two names, either Bax888 or SHP654. Those are old names left over from, this was originally Baxter, then Baxalta, then Shire, and now Takeda. So these are projects that have been going on for a while. But they've just recently published a study that might have a good clue as to what's going on with these. Under the column promoter transgene at the bottom there, you see it's B domain deleted and CPG depleted and codon optimized. What codon optimized means is that they've taken the gene one thing about a gene is that you can code for the same protein by several different methods. In other words, the language in the gene that tells you what amino acids to put in the protein. There are several versions for that. It's almost like you could have a gene in English or French or Spanish. But what codon optimized means is that the company has figured out which of these languages helps produce the protein most easily, so that the protein is the cell can basically produce more of the protein because it's an easier process. This has been important. This has been something that's been going on for a long time in producing recombinant proteins. Now, recombinant proteins aren't produced in liver cells. They're produced in giant bioreactors, usually in some kind of animal cell, in a pharmaceutical plant. And a pharmaceutical plant doesn't have an immune system to worry about. So in doing codon optimization, they've picked the best codons or the best language to produce this protein as though it were in a giant bioreactor in a pharmaceutical plant. What they've come along, and actually the people working on an earlier Baxter-Baxalta gene therapy that was discontinued by Shire because it was getting poor results, those people have gone back and looked at the results, and they figured out that this CPG sequence, you can see that there, that's a certain sequence in a gene that if that sequence is there, it alerts the immune system that this is something that it might want to get rid of. Turns out a lot of viruses and bacteria have CPG sequences in their genes, and that the body uses that to tag a cell that might be producing the product of that gene, and say, this is something that we got to watch out for, and we're probably going to go and destroy that cell. Well, it may be that in doing this codon optimization, they've inadvertently introduced a number of these CPG sequences in the gene, and that is what's causing the production of factor VIII to go down, because the immune system has been targeting those cells and deciding it ought to get rid of them. So you don't have as many cells producing factor VIII, so you don't have as much as high a level. Now, this is just kind of a theory right now, although there's a lot of work going on it, and it seems like there may be a lot to this. So let's go on to the next slide, please. All right, here's hemophilia B, and it's kind of the same situation. We've got two on top that are in phase 3 studies and getting very good results. And neither of these has really seen a drop-off in factor IX production. And then Freeline, a British company, has been coming up fast in their studies. St. Jude, I'm not really sure what they're doing at this point. They haven't had a lot of activity. And then Sangamo is interesting at the bottom there. It was discontinued, as you see. They were actually going to do gene editing. Instead of just putting a new gene in, they were actually going to edit the genes that are already there to put in their new gene into the genome on the chromosomes. that's a little more complicated than I'm going to get into right now, but it worked, but not very well. They got like, 1% factor 9 levels. So they discontinued that, basically back to the drawing board. They're working more on this because it's ZNF, you see there, stands for zinc finger. That's it's a kind of an enzyme that you can use for genome editing. And it seems like it has a lot of promise, but they just need to work on it again a little bit to get there. And at the top of the right-hand column, I've got clinicaltrials.gov. That's a website that lists all the clinical trials for all pharmaceuticals, all different kinds of drugs and other products that are going on. It includes all that are in the United States, and it includes most that are in the rest of the world. So if you ever want to find out about clinical trials or you're interested in participating in one, first talk to your doctor, but you can also go to clinicaltrials.gov and just see what clinical trials have been registered there and what might be available for you to participate in. And I'm not going to go a lot into participating in clinical trials, but a number of people in the hemophilia community think that's an important thing to do. A lot of the older guys are doing it, figuring that this is, kind of a legacy that they need to leave to the younger ones, is to try to help develop these products. None of these products could be developed if we didn't have people to test them in, people who volunteer. So then let's go to the last slide. Okay, these are all the different organizations that are working on gene or cell therapy for hemophilia. And I'm not going to go through all this, but my only purpose here is to show you that there's a bunch of them. And so what I'm thinking is that, and this is my own opinion, I should have said in the beginning that I give a lot of opinions as we go along here, but my opinions are my own and don't reflect either HFA or the Coalition for Hemophilia B. So if you don't like what I say, just blame me, don't blame them. You can see that there's a lot of people working on it in a lot of different ways. And so I think these first gene therapies that come out are definitely going to be game changers. And some people are going to want to go, right to them, and basically eliminate having to infuse all the time. But I think there are even more advances that are going to be coming along. That maybe 10 years from now, gene therapy will look totally differently than it does now. We'll just have to see. Again, with all of this stuff, it's all unknown, and we just kind of have to see what happens. Even though there's so many of these, they could all fail, or they could all be successful, and then you're going to have a hard time figuring out which one to use. So anyway, that's my last slide. Let's go flip the slides one more time, and I've got a slide for questions. And so if you have a question, I'll be glad to try to answer whatever I can in the last few minutes. And let me put your questions in on the chat screen. And let me get into it here. Yeah, researchers don't know what it's like to live with this bleeding disorder. I go along with that, not so much just with bleeding disorders, but I, for instance, am hard of hearing, and I'm trying to get new hearing aids. And I'm convinced that the companies that make hearing aids, that none of the people who design these things really use them. And they mostly just don't really do what you need them to do. Okay, learning more, listing. Okay, I'm not sure we have any real questions here. So if you do have a question, feel free to put it in here in the next few minutes. With that, I guess I'll just hang out here and see if there are any questions, or the moderator can take this back over and
thank you.
They need to do. It's been a pleasure talking to you, and I want to thank HFA for inviting me.
Well, thank you, Dr. Clark. We'll hang out just for a minute because been a little slow this week as questions have come in, but we'll give it just a minute to see if any come in. I always appreciate hearing about all of these products. It's illuminating every year as we put together the emphasis, or for this week, and it's not coming up because of my filter, but Dateline Federation issue with all of our the products. There's something new every year. And so it's really important for us to stay on top of these things. And we appreciate your stepping in this week for us and covering this very important topic. Well, I don't see any more questions for you. So we're both here in Colorado, I believe. And I will say I lied. There is a question. When and how will gene therapy companies know if there can be repeat dosing? What's your thoughts on that, Dr. Clark?
Yeah, that's a real good question. Actually, Unicure, who is doing one of the factor 9 gene therapies, seems to think that that's not going to be a problem. Most of these, everybody's worried about people who have pre-existing antibodies against the AAV viruses and whether those are going to prevent the gene therapy from working. But in Unicure, in their factor 9 gene therapy, have quit worrying about that. And basically, they've shown that with a lot of subjects who have actually fairly high antibody levels against AAV, that it doesn't matter. The gene therapy still works anyway. And at this point, we don't really know whether that's just a characteristic of AAV5, the subtype of AAV that they're using. A lot of people are also working on synthetic AAV viruses that aren't very immunogenic, that won't really raise an immune reaction. So I'm not sure exactly when they're going to know, but Unicure seems to think it's not going to be any big deal and that they could basically do it today. Whether the FDA will give them permission to do that is another story. They probably will have to do some more clinical studies on that. But then there are other approaches. You can actually do things like eliminate a number of the AAV antibodies. You can run your blood through a chromatography column that picks out the anti-AAV antibodies and then give somebody a gene therapy, and there shouldn't be any real immune response against that. Although we don't really know that for sure. One of the things we're finding out in all this immunology research that we've come up with from COVID is that the immune system is much more than just antibodies. There are lots of other cells that, even if you don't have antibodies, can go in and eliminate viruses or do whatever. The immune system just keeps getting more and more complicated as we go along. It's not that it's getting more complicated, we're just learning more and more about it. So I don't have a good answer for that. Unicure thinks it's no problem, you could do it today. I'm sure that they will all be working on this, especially for factor 8, where it seems like they might have to redose people. We'll just have to see.
Thank you. I mean, another question I think that comes to my mind, I'm a mom of an almost 18-year-old, and just, going back to your very beginning slides, there's been so much progress in that time. Is this normal? Is this normal for a specific range of disorders that there's this much progress in a 2-decade period?
I think it is normal, yes and no. I mean, this hemophilia is, even though we're a very small population, a lot of people are interested in it. Certainly the pharmaceutical companies are interested in it because there's a lot of money in hemophilia. But there's also a lot to be done. I mean, we kind of understand hemophilia better than we do a lot of other diseases. Although as we go along, we keep coming up with more questions. If you go back to that timeline slide, and you don't necessarily need to go back to it, but you'll notice that from the late 80s to the early, there was a long period of time when there was very little activity on factor 9, even though there was a lot of activity on factor 8. And I don't really know why that is. I guess they thought that they'd gotten to where they needed to be with factor 9, and factor 8 was really the larger population and needed more work there. But now they've come back to factor 9 and they find out that, in a way, factor 9 is easier to work with than factor 8 because it's a much smaller protein. So because of that and other reasons, they may be focusing more on factor 9 now. But there's a lot of activity just going on both sides, on both A and B. I kind of bemoaned for a while that there was not much going on factor 9, and that all the work was on factor 8. I don't really have that problem anymore. And a lot of these things, I don't think I mentioned it, but a lot of these things like the rebalancing agents and all work for both, not just factor 8 and factor 9, or not just hemophilia A and hemophilia B, but also they'll work for inhibitor patients too, because they don't involve factor 8 or factor 9. So that inhibitors to factor 8 or factor 9 won't really matter. So yeah, I don't know if this is, I don't keep up with a lot of the other disease states. I'm sure there's way more research going on in things like cancer and heart disease, and certainly on COVID, than there is on hemophilia. But there's a good amount going on in hemophilia, and I think we can assume that it will continue. So that's basically my prediction on that. Is that I don't know, but I think it'll keep going on. Here I see I have this another question from Steve Long. Once the AAV has delivered the factor-making mechanism, does it disappear, leading to no more immune response? Yes and no. Depends on, it disappears, but within a time frame. They've actually found that some of the AAV virus particles can stick around. They're eliminated from different body systems in different amounts. And I don't have just the differences in the tip of my tongue, but there's some that some of the AAV particles have lasted for up to about a year in some systems, but in others like saliva and urine, you see the levels go down very quickly. So it seems like most of them are eliminated pretty quickly, but there's a small amount that kind of hang around longer. And then he says, thus the success with some steroid treatments to get through the initial phase. Yeah, the initial phase, the initial immune response, I'm not sure if that's because, well, it's probably, of course, yeah, partly because of the AAVs hanging around, but it may also be partly because of other parts of the immune system that they get triggered, and it may actually be attacking some of the liver cells that have been transformed. I mean, this is part of it that we don't really understand well. We know that in some patients, they have elevated levels of liver enzymes, which indicates that there's some inflammation or some immune response going on in the liver, but other patients don't get that. They know that they can use the steroid treatments to get that liver inflammation down, and that usually helps to get higher factor 8 or factor 9 levels as the treatment goes along. But they don't really know for sure exactly what's going on and what the mechanisms are. They just kind of know that the steroids work and they have some ideas why they work, but there's still a lot to be learned there, I think.
And a final question, you know, our von Willebrand's friends, what is out there for von Willebrand's disease? You touched upon it lightly, but why is that so hard, or you see less research for it?
Yeah, you know, that's a real good question, because theoretically, there are a lot more von Willebrand patients than there are hemophilia patients. In fact, the estimate that everybody uses is that 1% of the whole population has von Willebrand disease of one kind or another. I think there's some different reasons for that. I probably should have included von Willebrand in this, and I know that there are recombinant products and there are plasma-derived products for von Willebrand, and theoretically, they work fairly well, but I haven't really talked to any von Willebrand patients to see if that's true. Von Willebrand factor is harder to work with. It's another giant protein. And there are a bunch of different types of von Willebrand disease. And some of them are very severe, and some of them, you know, almost don't make that much difference except in certain circumstances. But I don't know why there isn't more work on that. It's basically the same question to me as why isn't there more focus on women with hemophilia? In both cases, it seems to me like here's a big market, and why aren't people doing something about it? But I don't really know the answer to that.
I always appreciate a good I don't know either. So that means that it's all up to us to go find the answer. So, well, Dr. Clark, thank you so much. I was saying earlier, I know you're in Colorado as I am, and we're experiencing some dark skies. So I appreciate you hanging in there with us today and presenting as our final session of the day. So thank you so much.
You're very welcome. Actually, I've listened to some of the earlier sessions, and I thought this was a very nice program. So thank you.
Thank you very much. Well, as we start to wrap up here today, just a few more things and pearls of comments to say. But you know, we really started on Wednesday with the look at how do we as patients work with our doctors, work with our providers to make good decisions. And we talked a lot yesterday about what do all these acronyms mean, and they keep coming up in every presentation that we've heard over the last 3 days. We heard a little bit about why it's important to participate in clinical trials, and then today more about registries and what the future looks like. As I mentioned earlier, it's been almost 18 years since I came into this community, and I remember very well sitting in the Hemophilia Treatment Center for the first time, trying to first learn how to spell hemophilia, and then trying to figure out what product to use. It was like trying to decide between chocolate and a sour candy. You like both, but which one is the right one for me in this particular moment? And we didn't really know, as new family without a family history of hemophilia, my husband and I really didn't know what questions to ask, and we didn't even know what we were looking at. It's crazy to think that in that short amount of time, just 18 years, the products for bleeding disorders have nearly doubled, and that novel treatments beyond just clotting factor are coming down the pipe and are already here in at least one product and category. As patients, we need to know our options. That's our right, but it's our responsibility to go out and educate and empower ourselves to learn about those things. From the HFA perspective, it's been one of our founding principles since 1994 to give you those resources so that you can be an informed patient. We want to be able to provide you everything you need, and it's our mission to assist, advocate, and educate the bleeding disorders community. We hope that we continue to be a trusted resource to you, and we hope to see you again very, very soon. Thank you so much for spending the last few days with us. As I go on with the thank yous, I also want to thank our sponsors. We've mentioned them several times, but once again, CSL Behring, Genentech, Hemobiologics, Takeda, Sanofi Genzyme, and Spark. We really appreciate your faith in an inaugural event and stepping forward and helping us out. I'd also be remiss if I didn't thank the HFA staff and our board of directors, again, for their faith. It's a really amazing the amount of work that goes on behind the scenes to kick off an event, even a virtual event. And there was a lot of hard work, late nights, and just really appreciate everyone's support as we developed this program. I also, again, thank you. You've hung in there with us. I appreciate everybody sticking in and attending this week. If you wouldn't mind, take a minute as we close out the browser. There will be that same 5-question evaluation that you've seen over the last few days. Please take a minute. We really do value your feedback. And then the big one, save the date. Symposium is going to be a virtual event again this year, and it will be over the course of 10 days, so October 18th through the 28th. Registration is opening very soon. Be on the lookout. The email will come soon with the registration link. It'll be on our social media and on our webpage. So be paying very close attention. Registration will open very soon for that. And it'll be much in the same format that you've experienced over the last few days. And again, just final, thank you so much. We really appreciate your trust in us, and we look forward to a return to seeing our friends and family in person. But in the meanwhile, stay safe and continue to connect with us even through Zoom. Have a great weekend. Hopefully everyone has a great summer, and we'll see you very soon. Thank you.